Hi everyone, my name is Feng and I'm from the University of Michigan, and today I will present my work, GenomicKB, a knowledge graph for the human genome. Since the completion of the Human Genome Project, millions of studies generated data and annotations in different fields, such as genome, epigenome, and 3D nucleome. With all this data, we can answer a lot of questions of our interest. For example, if we are interested in how a genetic variance leads to a disease, we can follow this diagram to answer it, from variants to genes, and then to pathways, tissue and cell types, and finally diseases. Different data sources can help us connect these modalities together. For example, GWAS is the relationship between variants and diseases. And the relationships of variants and genes can be found on GTEx and GeneCode. And pathways contain relationship between genes, which we can find those data on KEGG and Reactome. And in the end, we have the ontologies for the relationships between tissues, cell types, and diseases. So to sum up, we have different relationships between different modalities, and the answers to our questions, such as how a genetic variance affects a disease, are usually the combination of these relationships, such as GWAS, EQTL, gene regulatory networks, and location and 3D interactions. However, it is very challenging to integrate this data for answering these questions of our interest. First, it requires table joining and coding skills, and second, we need to deal with the ambiguity, duplication, and disagreements among all the different data sources. Therefore, we need a method to seamlessly integrate this data together, and our solution is to use a knowledge graph to do that, and this is our work, Genomic Knowledge Base, or GenomicKB. So what is a knowledge graph? Knowledge graph is a machine-readable format used to organize and represent information. It connects real-world concepts such as people, location, and abstract concepts, and also their relationships to link and integrate data from diverse sources. They are good at data integration with flexibility and scalability, supporting reasoning and inference, and also facilitating machine learning and artificial intelligence. Therefore, we plan to build a knowledge graph like this. It covers different modalities, such as genes, variants, and diseases. They are connected with each other with diverse relationships. And what we need to do is to define a rule to convert the tabular data into nodes and edges, or we say entities and relationships. This is an example data from GTEx. This table shows two different variants, two different EQTLs of the same gene. It can be converted into a graph with 3 nodes and 2 relationships. All the nodes and relationships can also have properties. And in the end, we obtain a knowledge graph of more than 30 data sources and more than 300 million entities, more than 1.3 billion relationships, and almost 4 billion properties. With the knowledge graphs, cross-modality analysis is turned into a coding-free query process. For example, a user can ask which GWAS variants of type 2 diabetes are intragene variants, and whether these related genes express in pancreas. What are the gene functions annotated from gene ontology? Our query system will translate this query into the cipher query language for knowledge graphs and return the results as a subgraph like this. To support the user queries like the previous example, we built a web platform for it, and this is linked to our website. This is the front page of GenomicKB. It provides tutorials, some introductions, and documentations. We also provide some example queries which you can directly load into the GenomicKB query engine. So let's move to the query engine. To explore GenomicKB, let's look at a simple query. Suppose you want to know which sequence variants are located in the CTCF gene. First, you need to add 2 nodes for the 2 concepts, a sequence variant and a gene. You can add restriction to the node properties, such as specifying that the gene's name must be CTCF. Next, specify the relationship between the two entities. By clicking Add Edge, click on the Start node and drag your mouse to the End node. Now you finished adding the edge. The edge type must be selected on the left panel, and after creating the query, click Submit, and GenomicKB will guide you to the result page. The results are subgraphs from the knowledge graph that match your query patterns. You can move the nodes by clicking and dragging, except for the nodes for genomic regions. You can also click on a node to display the details. Sometimes the entire query result includes millions of nodes and edges, which might take minutes to export. Therefore, the result page only displays the first 5 to 20 matched patterns with a smaller subgraph to ensure query speed. However, you can always download the complete query result by clicking Export and leaving your email address for receiving the results. Our paper was accepted by Nucleic Acid Research, and you can refer to our paper for more details. And that's the end of my presentation. Thank you so much for your attention.